change. You're not going to hear much from me today. I'm just going to do a very brief intro and then hand over to Austen. But just for those that aren't familiar with EnviroDNA, we are an eDNA specialist company based in Melbourne, and we've just moved, it was now 6 months ago, moved into our new purpose-built laboratory facilities, which has been very exciting and a welcome change for us. And we work with a range of different organisations, from governments, not-for-profits, universities and R&D space, and private organisations and other consultancies as well, across Australia, and even a few international projects as well. And we apply this eDNA technology to a range of different areas as well, primarily in conservation, but more and more we're starting to work in biosecurity space, and we've been running a number of citizen science projects, which has been really great to get local communities involved in their local environment and start valuing and understanding more about mostly their local waterways. So as Jackie mentioned, we are talking about sampling methodologies today, and primarily we're going to be talking about the Smith Root eDNA sampling system. But before we get to that, I'm going to take you through the way that we've done it for a number of years, a manual sampling methodology, which you can see on the left of your screen there, and the Smith Root backpack system on the right. So this manual sampling methodology, we came up with a number of years ago. It's quite simple, easy to do, and doesn't require a lot of equipment. And we found it particularly valuable for our citizen science projects. So we're able to demonstrate the technique and train people with no previous experience very easily. And we can also sort of send these packs out to people via the mail, which is nice and simple. So rather than hear me describe it, I think the easiest way is for me to play this video, which will give you a very brief demonstration about how we do our manual sampling. So we certainly don't expect you to memorize that. If you do get involved in one of our projects, we have instructions and YouTube video. Oops, I don't want to watch that again. It's just to demonstrate the simplicity of the technique. Basically, each of those kits has all the equipment that's required for sampling a site, and we can just send out as many kits as required for projects. So just to highlight the sort of older style traditional methodology, and now I'm going to hand over to Austen to talk about the Smith Root system and a bit of the history and development of it, as well as the practicalities of how it's used. So I will stop sharing and hand over to you, Austen.
All right. Start this thing back at the beginning. Okay. Are you seeing my presentation? Let's try this one more time.
No.
There we go. Okay, how are we doing? Looking good? All right. Well, hey, yeah, I just wanted to start by saying, thank you for the opportunity to give this presentation. This is a system that we've been working on for a number of years now, and we're really excited about it, and we're excited that other folks around the world are starting to use it. And I guess without further ado, I'll kind of introduce Smith Root. So Smith Root's been around since actually the 1960s, and they're best known for developing backpack electrofishers. So going out into the water, creating electric field in the water, stunning fish so you can do fish inventory. So Smith Root's been doing, ecological sampling for a very long time. And so my role was, when I came on in 2015, was to see, what could we do in the environmental DNA space? Because it was still pretty new at that point, and there weren't really any commercial labs, there weren't really any commercial systems. And so we started out just by seeing what people were doing at the time. And the methods were kind of broken down into, two broad categories. You have those who were, collecting water in the fields, with Nalgenes, sterile Nalgenes, and taking that back to the lab for filtration. And then there were a couple of really good protocols that came out about that time. One from the USGS, so this is the Laramie et al. Protocol here. They showed a couple of different methods for in-field filtration using these Nalgene filter funnels that have 47 millimeter membrane filters in them. And they were using a variety of different, pumps like drill-powered peristaltics to filter that water and then fold up those membranes and stick it into some kind of preservative like ethanol or Longmire's buffer. Another great protocol from that time was the Karim et al. Protocol. Another USGS, or actually the National Genomics Center group, came up with this protocol. So they were taking these geopumps, another peristaltic option, adding those filter funnels. They swap out the membranes in those filter funnels for a glass fiber because it gets a little better flow rate. And then they send this out to a whole bunch of project partners. So we started looking at, what are the limitations of this? And, we've heard a little bit about syringe filters already today. So the issue with water transport is that it really limits your sample size. There's only so much water that you can tote back to the lab to filter. There's also the problem of DNA degradation. So if you're bringing water back to the lab to filter it, there's always some transport time, and there's the concern that degradation occurs in that interim. The pore sizes that are available in most of these things are also not the best for filtering eDNA. If what you're interested in is fish or other vertebrates, the particle size tends to be a little bit larger. And so if you're using something that's designed for filtering microbes, you're really kind of choking down that filter and really limiting the amount of water you can filter. And then that sampling gear was not purpose built, right? Like it was, designed for other processes, and so therefore it was not made for people who are doing eDNA sampling. So this is what we conceived, was this fully integrated backpack eDNA filtration system that contained purpose-made single-use inline filter housings that allow you to then get away from the location that's being sampled, right? So you don't have to go into the water to collect that sample. There's no contamination risk associated because you have a pole extension. It would then have a smart pump, so a computer-controlled pump that's going to be integrating sensor feedback and controlling all the filtration parameters. A remote control so that a single operator could be running one of these things in the field. And then a place, storage for all of those consumables. So all those single-use filter housings that you would need. And this is a picture of the system. So I don't know if anybody hasn't seen one of these things before, but there's starting to be quite a few of them now out in circulation. So, please let us know if you want to try one out at some point. We'd really be excited to show one to you. So going back to kind of those custom-made eDNA filter housings, why those are important for this process. We have a mechanical engineer on staff who really started from ground zero with the thought of what are you doing with eDNA filtration? And he created a filter housing based on this high pressure, low velocity principle. So you can see that this filter housing, as water is going up, it rapidly expands into this chamber here. And what that does is it slows down the movement of those particles. So one of the biggest problems we have with eDNA filtration is clogging, right? Like everybody's dealt with clogging. So this is designed to take some of those larger particles and allow them to kind of stay in suspension while the smaller particles are basically being enriched on the filter membrane. And so for people who have used these things, you know, sometimes they'll take a filter funnel and they'll take one of ours, the same membrane, same pore size, same everything, and they're getting more water filtered through the Smith root filters than they are through a funnel because of this principle. The other beauty of it is that we can load it with really any membrane material. So we package these at Smith root. And so we're, you know, if people are saying, hey, I really like nitrocellulose as a membrane instead of polyether sulfone, or I really want glass fiber, or whatever it is, we're able to do custom runs that way for folks. And we're also able to, you know, have stock items. So I think right now we're stocking 4 different pore sizes of different materials, so that we have, you know, some that are readily available for a range of different habitats at any given time. And, you know, this design allows you to concentrate a larger volume of water, which therefore improves detectability. So those filter housings come packaged in these little filter packets, and the filter packets contain the housing with a 47mm membrane filter. This little tubing extension, sometimes we'll call it the snorkel tube, that attaches on here to give you about, you know, 6 or 7 inches to get into the water that you're sampling without submerging the filter housing. And then the original packets also come with sterile forceps, so that you can fold up that membrane in the field and stick it into sort of a DIY preservative, if, you know, because we found a lot of people were doing different things for preservative. I have a lot of experience using ethanol. Another great option is silica beads. So just using a desiccant, if you submerge, you know, fold that filter up, stick it into a whirl pack with a desiccant, that's a great storage mechanism. So now going back to the pump system. So we've thought about what are the important things to measure while you're doing filtration. And one of the ones that we thought would be really important is to use a pressure threshold. So basically a programmable pressure threshold, you know, which is something that the system will not exceed, and it's user programmable. The reason we use a pressure threshold is because primarily what we're doing is we're filtering epithelial cells, right? If we're going out there and we're you know, looking for fish, mostly what we're detecting is sloughed epithelium. And those cells, if they are basically experience too much pressure, could potentially lyse, and then the DNA that you're trying to capture would actually slip through the membrane itself. So we thought that it would be really important to include a pressure threshold, because the oceanographic studies that have been doing this a lot longer than us recommended it. So the other thing, that kind of key component of this system, is a flow meter and the ability to program a bunch of parameters around flow. So the obvious reason to have a flow meter in there is to just be able to meter the volume filtered. So this is, you know, this system has theoretically no limit to the volume you could sample, right? So if there are very, very, you know, few particles in there, you could filter tens of liters of water prior to the filter clogging. Like I've taken 10-liter samples on a single membrane before when we were filtering well water. It also gives you the ability to set a target flow rate. So one of the reasons why flow rate has become really important is that depending on the level of suspended particulate in the water, you can actually increase your filterable water volume by slowing down the speed of filtration. So what we've been recommending to folks is that if you're working in turbid waters, try dropping your flow rate down to around 0.5 liter per minute, or maybe even a little bit lower, to maximize your filterable water volume. And then if you're working in pretty low particulate water, you can crank that thing all the way up to 1 liter per minute or 1.5 liters per minute and really just get the job over with quickly. And then the other piece that has been really exciting for folks that we didn't really anticipate was going to be a major selling point is a filter clogging alert. So it tells you when the filter is clogged, right? So when flow drops below a threshold value, it's going to beep at you and say, hey, your filter's clogged, change out filters, do something different. Because prior to this, people were, you know, so fixated on getting, say, a 1 liter sample, and they were pumping their, you know, 0.45 micron filter with their peristaltic pump, and they were just sitting there for half hour waiting for the last 10 milliliters to go through. That's not a great sample because there's a potential for degradation there. There's the potential for the cells to experience a lot of pressure for a long time. So having the ability to have the system tell you, okay, it's time to move on, an objective measure of that is really useful. So we wrote up a system description for this and submitted it to Methods in Ecology and Evolution, and their response was, that's really cool. Now tell us how to use it. So we ended up doing some experiments, and those are contained in this paper here. So this contains a description of the system, as well as some of our kind of range finding of what parameters we should use in different scenarios. So I'm not going to belabor that too much. The one thing I am going to touch on is that the system is very fast by comparison to a lot of other systems. And so this is looking at volume filtered and filtration time over a bunch of different samples. And on average, you can see that 2 liters of water can be filtered in about 3 minutes. And that's end to end. That's from the time you push the button to the time that the sample is fully preserved, is about 3 minutes to collect a 2-liter sample, which is pretty exciting for those who'd been thumping along with peristaltics. And this, I believe, is going to come through to you as a little bit of a choppy series of images here, I think because of the lag, but I just want to illustrate other things that you can do with a backpack filtration system. We're now integrating the use of multiple filter housings onto the end of those poles. So we've got a new pole here that we're calling our trident pole, so you can put 3 filters on there simultaneously. It also gives you the ability to do a mobile transect. So you can walk along and filter and try to work into some of those down woody debris habitats or some of those side channels that maybe you weren't hitting, and create a spatially integrated eDNA sample. You know, one of the biggest challenges with eDNA is that it's super heterogeneous in space. And so the ability to do a spatially integrated sample means that you might be able to do a better job of representing what's in that habitat with a fewer number of actual filters. Another really cool thing that this system has is a GPS on board. So if you are doing mobile sampling, it can do a little snail trail of the, you can create a map with the GPS data of where you filtered. It also, even if you're doing point sampling, so this is actually an image taken where we were comparing mobile sampling to point sampling. So you can see we've got GPS locations from 3 point samples, the bottom, middle, top of the transect, and then volume filtered is here mapped in color. So you can see that the volume filtered accumulated over the course of the transect. So that's a pretty slick feature for those who are interested in kind of more advanced sampling options. The other really cool thing that this system does, and by having that GPS on board, is that you could standardize your volume per unit distance. So because you got a flow meter and you got a speed, you can get a milliliter per meter value. And so say you went and you filtered at 50 milliliters per meter 1 year, you could go back and filter the same transect at 50 milliliters per meter a 2nd year. And that allows you to standardize your mobile sampling. This is just an example of one of the CSV data records that the system spits out. So it's going to be logging every 2 seconds all of the parameters that the system is measuring. So you're going to get, volume, pressure, flow rate, all that good stuff, as well as some sort of summary data here at the top of the CSV. One thing that's not quite out yet is our eDNA data viewer. So we're working on a software tool that allows you to essentially click through one of those individual samples. So each sample is going to get its own CSV file. And you could click on one of those sample CSV files and quickly visualize that data record. So say you got a weird result you weren't expecting from a big data set, you could go back and quickly review, did something wonky happen with that particular sample in the field? Okay, so now segueing away from our backpack sampler, I want to talk about one that is getting a lot of attention right now, which is our eDNA citizen scientist. This is a kind of a smaller scale, lower cost version of a filtration system. It originally was modeled after, I don't know if anybody saw like the hand pump vacuum flask filtration model. So people were going out with a sidearm flask, plugging into a hand pump, pulling a vacuum on that hand pump, and then sticking some sort of a filtration system into the water. So my thought was, well, can we just make an analog of that is battery powered, right? So it'd be lightweight, super portable, lower cost than our fancy computer controlled pumps. And this is what we came up with. So it's limited to a 2-liter filtration volume. So you've got an air pump in here that's pulling a vacuum on this chamber, and that vacuum is being conducted through to our filter housing there on the end of the line. So once this water fills up to almost a 2-liter mark, you invert the filter, you do the line clearing step, and then you've got yourself a very fast 2-liter sample at a fixed pressure of 10 psi. Some people do actually use these for, they'll empty the flask out, and they'll reseal it, and they'll go, continue filtering. So you can do, 4 6-liter samples on the same membrane if you can get it to reprime. Sometimes after it's already been kind of clogged up, it's hard to get it to reprime. But this researcher here, Bettina Thallinger, uses this pump for a 4-liter sample pretty regularly. Okay, and then the other thing that we're really excited to talk about today is our self-preserving eDNA filter housings. So this has been, of all the things we built so far, this is probably the most successful. So what we did almost by accident was to create a filter housing that preserves the DNA on the membrane by rapidly desiccating it. So we were experimenting with different biodegradable plastics, biodegradable resins, and discovered that one of them is highly hydrophilic. And so if you injection mold that resin into the bottom half, this green half of the filter housing, what happens at the end of filtration is it just wicks the moisture out of the membrane almost in seconds. It's really, really quite rapid at pulling the moisture out of this membrane to create rapid preservation. And the reason why desiccation works so well as a preservation method is because the goal is to basically prevent enzymes from breaking down the DNA. So if you remove water, and ethanol does the same thing. It essentially removes the water so the enzymes can't function. They can't break down that DNA. So this filter housing does the same thing. So basically you take your sample, you invert it, you allow the pump to run for about 30 seconds to air dry that membrane, and then it just goes directly back in the bag. So you don't even have to touch, you don't have to open it. You don't have to introduce the possibility for contamination in the field. So when you're done, like I mentioned, the filter just goes directly back in the bag. You can throw a label on it, and then it's just room temperature storage. So you stick it in a cardboard box, shove it on the shelf, and then you can wait to process it whenever you have the opportunity. So there's no membrane transfer step, which reduces the potential for contamination. There's no cold storage required, so you don't have to be lugging cold storage or preservation buffers into the field, and it reduces the overall per sample field time. So you're not having to mess around with those filters, those membranes in the field, which speeds things up. If anybody's interested in the details of those, so we did a pretty lengthy test of those filters in terms of their ability to preserve DNA over time. And again, I won't spend too much time on this because I don't want to run us out of time here. But we did a whole bunch of samples from the same environment that contained New Zealand mud snails. And we extracted those samples at various time points. Half of the set was self-preserved, half of the set was preserved in ethanol. And what we saw over the course of 172 days, so roughly 6 months, was that there was actually slightly higher eDNA recovery from those self-preserving filters than there was from the ethanol preserved samples. And really no significant signs of degradation in either set after 6 months of room temperature storage. One really interesting result from that study, and I'm a little bit embarrassed by the sample size, honestly, that we published here, but we had a very limited number of them to work with at the time. But we compared, the reviewers really wanted us to go out and show them, a field example of using these things, not just a laboratory study. And so we compared detection of, I believe it was a frog species, spotted frog, using ethanol as a preservation method and using self-preserving. And we found that on average for the field samples that we were getting, oops, yeah, for some reason that didn't click through, but roughly 2 times the amount of DNA on the self-preserved samples as we did in the ethanol preserved samples for that particular field trial. I don't have a good explanation for this. Them's the data. So, but I guess suffice it to say that the self-preserving filters performed quite well. And then lastly, I'm going to segue and just talk about some of the exciting new developments that we're working on right now at Smith Root. So everything I've talked about up until now, that's stuff that we have on the shelf. You can buy it from EnviroDNA. You can consult with us about, different sampling protocols, different approaches for your particular sampling habitat. This is stuff that was kind of, this is all the R&D. This is the stuff we're working on, the cutting edge. So one of the things that a lot of people are interested in is autonomous sampling. And so we are developing an eDNA sampling robot using our self-preserving filters because it really simplifies the whole automated sampling process. So there are a few different automated samplers out there right now. Most of them are really expensive and quite complicated to run because they're doing either microfluidics to do automated extraction in the field or automated preservation using liquid buffers in the field. Our samples, you just have to pump air through it, right? So it filters water, and then you run some air through it at the end, and you're done. So this system here is a prototype that we've developed. Instead of a plastic bag housing each sample in this case, it's an aluminum manifold. So each one of these filters is going to be in its own aluminum chamber, and then it's housed inside this box. And then you tell the sampler, okay, I want you to take one sample, every other day for a month or whatever it is. And then the system goes to sleep. It goes into a low power state. And then at the scheduled time, it wakes up, filters the water that you want, shuts itself off, and then waits for the next sample. And the target cost for these is probably going to be somewhere in the ballpark of around 20,000 USD by comparison to 100,000 plus for some of the other systems that are a little bit more sophisticated. And then the last thing I'll touch on is kind of the polar opposite of that. Instead of going fancy, we're talking about trying to find ways to use those self-preserving filters to do manual sampling. So it seems like the syringe filter model is very popular, and the reason why it's popular is because there's no expensive pump. Anybody can do it, and you can make a little kit, and you can send lots of those kits out to a lot of people. So we identified a really low cost pump system. So this is a manual pump. It's like a transfer pump for transferring oil or gasoline from one location to another. It's just a pump with a one-way valve. So it's essentially it's a syringe filter, or it's a syringe with a one-way flapper valve on it. And it allows you to filter potentially an unlimited sample volume, right? So instead of having to reload that syringe each time, you just keep pumping that thing and it'll keep filtering water. And so I have, I don't know if anybody has seen on Twitter, if you check us out, we have an example of filtering in 2 minutes a 2-liter sample using this approach. So it's something we're looking into a little bit harder right now, whether or not we should be putting these things on our website for people to buy. With that, I'll stop talking and we'll take the questions, because that's the exciting part.
Thanks, Austen. I think anyone that's used the syringe to do a 2-liter sample would be very interested in that pump system.
Great.
All right, so I should have mentioned at the start to add questions to the chat system. So if you do have questions, please add them to the chat and we will run through them. Jackie, have you been monitoring the chat?
Yeah, we've had a few come through. Sorry, I've lost my ability to turn my video on, so I'll just operate like this. That's all good. So we had a question come through. It says, what is the best sampling method for high turbidity water, characteristic of many Australian dryland rivers, where due to colloidal suspended clay particles over 1,000 NTU is not uncommon?
Wow.
Yeah.
Yeah. So, I mean, typically my response to that, and I don't know, Josh, do you want to jump in here, or should I?
No, I think it's a question we get a lot, and I haven't come up with a really good solution. And I think you had some really good tips in your talk. So yeah, I'll leave you to it.
Yeah, usually what I tell people to do is to, first increase your pore size. So I don't know what this particular user is doing in terms of filter pore size right now, but we are going with our kind of primary recommended pore size is 5 micron, which sounds really big. Like if you've been working with 0.45 micron or even 1 micron, it sounds kind of scary to go to 5 micron because, what are you losing? What we have found over time is that the increase in filterable water volume offsets any loss that's actually going through the filter. So you actually have higher overall detectability if you maximize that filterable water volume on a 5 micron than if you say, try to just go with a 1 micron and slam through as much of that water as you can. It's also supported by some of the size fractionation studies that have come out. So the most famous one, and I can show you here. So this Turner paper is now it's pretty old at this point, but they looked at a bunch of different filter pore sizes, and they were doing serial filtration, right, or size fractionation filtration, where you pour in through the largest pore size and it goes through a series of smaller and smaller pore sizes until you do precipitation at the bottom. And they found that the majority of the target DNA they were interested in, which was fish DNA, was on the 1 micron pore. They actually didn't test anything between 1 micron and 10 micron though. And so we have done, since then there have been a lot of studies, including our own, which shows, so here you've got filter pore size 5 micron versus 1 micron. And we increased the pressure and stopped filtration with our system once it hit basically low flow. So once it hit clog point. So we maximized the filterable water volume. And we actually got more total DNA on those 5 micron filters than we did with the 1 micron filters when we maximized that filterable water volume. And since we published this paper, and several other folks have also, Karen Goldberg's lab, I think, is also using pretty much exclusively 5 micron filters these days, because she also works in really turbid systems. So that's number one, is increase your pore size. And then the other thing is with our computer control pumps, like I mentioned before, slow down your filtration rate. So if you're just slamming those particles up against the filter at high velocity, you're kind of reducing your overall ability to filter water. So using the Smith root filter housing that has that inverted orientation and sort of does that particle enrichment, slow those particles down so that you can kind of get some of the smaller stuff without clogging up with the bigger stuff.
Yeah, and just to add to that, I mean, the other option is obviously do that to start with. You can increase your number of samples per site as well. I mean, the reality is your filters are going to clog up. You're not going to get massive volumes in these systems. And, our experience with sort of trying to pre-filter that is really difficult, because some of that colloidal stuff is such low particle size that by filtering that out, you're going to filter out your DNA as well. So, yeah, I think that those are really good tips. And to back that up, we've done some initial, some preliminary comparisons between the 5 micron and our syringe filters, and we're certainly seeing higher DNA yields from the 5 micron filters. So whatever you are losing in that small particle size is certainly being offset by the increased volume.
Okay, cool. We've got another question here. Someone says, I'm curious about the citizen science sampler. What is the expected filtration rate in turbid water?
Yeah, so here's the interesting thing about the citizen science sampler. Maybe I'll just click back up. We can look at that while we're talking about it. So this system, because it's an air pump, it's not a water pump, it just goes straight to 10 psi. So the filtration rate is whatever is possible at the membrane interface at 10 psi. So it's really dictated more by the membrane you're using and not the pump in terms of what that filtration rate is. So I've timed it a few times with our larger pore size filters, and we're getting filtration rates of like 1.5 liters per minute. Basically, you're maximizing the flow rate of those 47 millimeter membrane filters. It's a fast little pump. Yeah, so I think that's probably the best I can do to answer that question.
Okay, cool. We've got another one here that says, I'm assuming any data produced from the eDNA sampling with the backpack prior to data viewer will be able to be viewed using the viewer once it comes out.
Absolutely. Yeah, so it will be backwards compatible in terms of our ability to look at those files. So right now, the way that the system works is when you plug in a USB key, there's a database file on the system, and the computer on board actually looks at that database file, and then it exports a CSV file for every individual sample ID. The data viewer is just going to operate with that database file. So it's going to be completely independent of whatever the file format was that we're exporting right now, as long as it's compatible with the previous version of the database, which it will be. So yeah, I don't know if anybody's using our backpack pumps right now and sort of manually looking at those CSV files or come up with your own R scripts for visualizing them. We are hoping to make that a little bit easier for everybody.
We've got a question here from Reese saying, the first I've seen of the autonomous sampling work. In terms of the Smith root backpack, autonomous sampling, will the future direction of these products be making them more compact and portable? That would be amazing.
Yeah, great question. So the biggest complaints about the current backpack system is that it's still pretty heavy, right? It's like about the same weight as a backpack electrofisher. So it's it's kind of heavy. It's a little bit uncomfortable to wear. Those are things we're very aware of. We are looking at an alternative, and the time horizon on this is like 1.5 years or something like that. So I don't think this is coming out tomorrow, but basically we're going with a different version of the pump system, or the pump that's on board. So it's going to be much lighter. So the pump that's in there right now is a Hurricane water pump. So it's got a really big electric motor on it. We're going to a pump that actually has a higher priming pressure. It has the equivalent total flow rate, but much better air drying capacity. So it can really move air and water quite efficiently. And that air drying efficiency is really important for our self-preserving filters, right? Because you've got to maximize the amount of airflow across it to minimize the amount of water on it, so the preservation is rapid. So that's kind of step one. The other thing is we're looking at getting rid of the Hoffman case, which is that fiberglass box, which adds a fair amount of weight, and going to an aluminum housing. It's not an electrofisher, so we can get away with having aluminum housing instead of fiberglass. And then we're also looking at reducing weight in the dry bags. The dry bags aren't the lightest things in the world. So, reducing weight, making it more compact, those are definitely high priorities for us for the next generation of the system. The autonomous one, the picture that you saw, that's really just a prototype. That's not actually our production-oriented version. We are, as of July, going to have what we think is kind of the production version. It's going to be about half the size of the one that you're looking at, that you saw previously. I guess we can show it. Yeah, so the box will be about half this size, and the eDNA interface, the screen, all of that is going to be on the unit. So it's not going to be two boxes. It'll be a single box, that's about half this size with the screen and everything integrated.
Thanks, Austen.
Super exciting. I can't
wait.
Thanks. I'm super excited about it too. It's fun to work on.
So just as a follow-up question, Reese has also asked, for high turbidity, could a solution be an alternative extraction such as ethanol precipitation? Or is that not terrifically practical in this context?
Yeah, no, that's an interesting question. So I don't know if they're still doing it, but I think the U.S. Fish and Wildlife Service, the folks who do all the invasive carp work, they do centrifugation. So basically they take a bunch of falcon tubes, like 50 mil falcon tubes, and put them into a centrifuge and spin that water, pellet it, and then they're extracting the pellet out of the centrifuge water. So that's been working well for them because they were dealing with really high turbidity water. They developed that protocol before our systems were available and before anybody was using 5 micron. So I don't really know how the two stack up, next to each other, but that was their solution.
Although presumably, again, you're then reduced to transporting large volumes of water across the country.
Right. You're schlepping a bunch of falcon tubes full of water around.
Yeah.
And actually, I think they have a mobile lab, basically a trailer that they tow around with a centrifuge in it for doing this stuff.
So I've got time for a couple more questions. One from Maggie. She says, have you compared eDNA concentrations found on backpack versus citizen science sampler filters when filtering the same amount of water? Are they comparable?
Interesting. No, I haven't. That'd be an interesting study to do. I mean, in theory, they should be identical, right? Because it's the same volume of water through the same design of filter. It should be identical. But there are some slight differences in pressure and flow rate, so there may be some differences there. We haven't done that study.
This might be one for you, Josh. Dane has asked, with the eDNA syringe filters, you place the red cap from preservative solution onto the top of the eDNA filter. Does the empty small syringe go on the bottom of the filter or nothing before transport?
Yeah, and unfortunately it wasn't actually included in that video, but yeah, when you add the preservative to the filter, you leave that small syringe attached on the bottom, and then you cap the other end of the filter with that little red cap. And essentially what it's doing is it's sealing that preservative into the system and keeping the filter bathed in the preservative. And that's fine at room temperature. We've tested it up to 3 weeks with pretty minimal degradation over that time.
Okay, perfect. Well, we've only got 1 minute left. So that might be it for the Q&A today. But thank you everyone for joining us. And we hope you found this webinar valuable. And if you've joined for the other sessions, we hope you've enjoyed those as well. Just like all of our other sessions, we'll have a recording available if you want to catch up on this or share it around. And if anyone is interested in finding out more information, you can jump on our website and get in touch. We'd be happy to have a chat with you about whether it's Smithroot or some of our services. Yeah, so that's it for today. Thank you. And thank you everyone for joining. And thanks Austen for jumping on the time difference.
My pleasure. Yeah, thanks for having me.
Yeah, it was great to get particularly the background of the system, which I wasn't aware of, and some of that more technical details, which I think allows us to sort of tailor the sampling techniques a little bit better. So it was great to get your experience and knowledge, Austen.
Fantastic. Yeah, my pleasure. Thank you.